Thank you, Dr. Cox. Desiree is a force of nature. Also good to meet Colonel Strong. Hua. You know what I need? I need the little advancer, the little slide advancer. So, as Desiree mentioned, I currently serve as the CEO of a company called Cellularity. Thank you. Cellularity was created when we spun the cell therapy division out of a company called Celgene. Celgene was a $110 billion biopharmaceutical business focused in cancer and immunology. The cell therapy division there was created when they acquired a company I founded called LifeBank Anthrogenesis. That company 20 years ago decided to explore whether or not the leftovers of full-term healthy pregnancies might be useful as therapeutics. And we've made some big advances. I'm going to talk about it today. And I want to also say that what we once thought was regenerative medicine is actually the platform from which all of the breakthroughs in oncology and immunology are occurring, things like CAR T therapy, immuno-oncology, et cetera. I want to start off by making a little bit of a plea to be thinking about things in this sense. You know, the pace of progress in medicine is, when I'm out socially, is sometimes perceived as very discouraging. I'm often asked, how come you guys haven't cured cancer yet? How come my mother with MS is still suffering, and so on? And it dawned on me that in my other world, which is aviation, that the pace of progress in an industry, a technology industry, that one could argue could serve as a model for healthcare was really remarkable. If you think about it, from Kitty Hawk to landing in the moon, to see it Tranquility, took 66 years. Wow. So from flying a balsa wood box covered in fabric to putting two men on the surface of the moon, that occurred in 66 years. Stem cells were discovered about 60 years ago. Till and McCulloch in Canada stumbled upon stem cells. Does anybody know how they found them? They stumbled upon stem cells because they were looking at the effect of radiation on bone marrow, and they couldn't kill it all. And that's how they found out that there were these cells left behind that could actually continue to regenerate and rebuild it. So that remarkable progress in aviation occurred, in my opinion, because we don't have test pilots in medicine. Okay? In aviation, every time an aircraft was taken to its performance envelope, some bold guy, crazy guy, got in the cockpit and pushed it beyond its limits. Okay? That's how we broke the sound barrier. That's how we flew into extra atmospheric space. That's how we started flying at altitudes where there wasn't breathable oxygen. That was all because of test pilots. And I think that medicine could benefit from the psychology of the test pilot. And the reason I bring that up is I'm about 5 days out from having received an infusion of placental stem cells, and I take credit for being the guy who discovered these placental stem cells, and also had them injected intra-articularly in my old joints, my aging joints. And I can tell you that it feels really, really good to be able to claim wholeheartedly that I see myself now as a test pilot as well. So, I'm not going to belabor this. Everybody in this room knows what a stem cell is. The unique ability to replicate itself and then turn into all the specialized cell types through a process called differentiation gave rise to excitement that this might be a platform to treat all kinds of diseases. Cellular therapy, the concept of augmenting a system by restoring the population of stem cells that could in many cases repair a defect or augment a function like immunity, is the basis and the hallmark behind things like immuno-oncology, using engineered immune cells to combat cancer and so on. The bottom line is that all of us here recognize that the challenges in turning living cells into medicines really fall into some fundamental principles. You need to be able to source the cells. The attributes of those cells have to meet the biological requirements of the therapeutic. In order for this to really have a meaningful role or impact in medicine, you have to be able to deliver it to the masses. That means it's got to be scalable, and scalable with the quality that you would find in a conventional pharmaceutical. And then what we're seeing now, what we're experiencing now in the world of immunotherapy for cancer is that economics matter. Does anybody know what the current cost of T-cell CAR T therapy is? It's about $500,000 a treatment. Okay? That's unsustainable. That is unsustainable in any healthcare system. I don't care how good it is. At that price, you'd have to ration the therapy or ultimately dismiss it. That's tragic. So we, about 24, 25 years ago, when I was still a neurosurgeon at Cornell taking care of head and spinal cord injury, I was fascinated by the concept that stem cells might allow me to turn back on the regenerative properties of the brain to fix people I kept alive but were left with really, really significant neurologic deficits. I give my older daughter, who's 28 now, and she's doing her MBA at Wharton after graduating from Princeton and Oxford and Georgetown with all kinds of degrees. She keeps going to school, and I just found out that after her MBA, she wants to go to medical school. When she was in utero, was the first time that I noticed and recognized that the placenta, which I thought was the vascular interface between the maternal and the fetal system during pregnancy, was actually a factory. It was a stem cell factory. And then based upon that observation, I decided that I was going to go steal placentas out of the L&D suite, take them back to my lab at Cornell, and dismantle them. In doing so, recognize that the organ, the placenta, doesn't look like a vascular interface. It looks like a bioreactor. It's a very, very complex, cavernous organ where you've got little islands of tissue densely vascularized, and that's where stem cells take on the role of differentiated cells and traffic into the developing fetus. So, first and foremost, there's some properties of the placenta that made it the ideal place to find a therapeutic platform. That unique immunobiology of the placenta. You know that a mother carries a fetus and its placenta for 9 months. Even though she's only a 50% donor of the genetic material that makes up that newborn, that fetus, she doesn't reject it, it doesn't reject her, right? Pretty remarkable. Well, think about surrogate pregnancy. In surrogate pregnancy, a mother carries a fetus and its placenta for 9 months. She ain't even related to it, okay? She's not matched at all to that newborn, to that fetus, yet she doesn't reject it. You couldn't put an unmatched organ like a kidney into that maternal system and have it accepted like that. The other thing is that the placentas are abundantly available. You know, we're the only species, the only mammalian species that doesn't recycle the placenta generally. Most other species actually ingest the placenta postpartum. Which means that 130 million placentas are disposed of every year in the world. In fact, most places pay to dispose of them. Hospitals in the United States have to actually pay to have them incinerated. So a tiny, tiny fraction are utilized for one purpose or another, which makes them a great raw material. And in the hands of the right operators, you can produce lots and lots and lots of therapeutic products from one placenta. So our vision at the company that I founded was to find a way to harness all these unique biological properties of the organ, play a role in evolving the understanding of how cellular medicine could play a role in healthcare, and then to develop technologies which turn these living cells into deployable products that could fit into the conventional healthcare system. And the bottom line is that the company Cellularity, which is a successor to what gave rise to many, many CAR T programs still remaining back at Celgene, put ourselves in a unique position to advance the technology into the broader areas of what regenerative medicine hopes to be, what immunotherapy hopes to be, and what I believe in the future will be the preservation of human performance as we age. So in order to do this, we had to figure out a way in a reliable, consistent, reproducible manner, procure these raw materials under very, very rigorous controls to make sure that we didn't bring in any bad stuff, and then isolate, characterize, manipulate, expand, manufacture the cells, and then innovate those cells into product forms that could be used to treat different diseases. So from the placenta, over the last 20 years, we've developed a range of therapeutic products that have been used in human beings. Everything from novel immune cells of the placenta to placental stem cells, which behave a lot like what's been described for mesenchymal stem cells, stromal-type stem cells, but they have these unique properties afforded by being out of the placenta. In addition, you guys are hearing a lot at a conference like this and elsewhere that there are other byproducts of living cells that may be as valuable therapeutically, things like exosomes. And I'm here to tell you that we've been producing exosomes as a byproduct of our manufacturing for more than a decade. In order to make the economic sense I alluded to earlier, we had to come up with a way that you could spread the cost of that rigorous procurement, collection, databasing, and ultimately manufacturing across more than a single product from a single donor. And I used to call that the pharmaceuticalization of cell therapy. And what that simply means is how much can you get out of one placenta? I'm here to tell you that with our first product candidate, PDA001, we're able to produce hundreds of thousands of doses of cells from a single placenta. In our current programs in immunotherapy, where we've isolated a unique immune cell from the placenta, and I'm going to talk about that in a second, we can produce hundreds of doses per placenta. So that scalability is fundamental to meeting those requirements I alluded to up front. So from the placenta to immunotherapeutic products, so let's first talk about using the placenta as a source of materials that could be used in immunotherapy, specifically immunotherapy of cancer. From the organ, we had to develop a system that was reliable, reproducible, and in essence proprietary, so that it made business sense for us to invest. And that platform, which we call Impact, does a couple of things. It creates a very, very standardized process to get the raw material to a point where it's qualified to go to clinical grade manufacturing. The second thing is we had to find a way to make this sensible for the clinical world. And in my mind, what's sensible is the product has to be off the shelf and one size fits all. So what do I mean by that? Off the shelf means that the living cell product has to be in a stable, inventoriable form that has a long shelf life. You guys go to the store, you look at expiration dates, right? Anybody have an idea what the expiration date for a cryopreserved living cell is? It's probably infinite, okay? Probably infinite. Hal Broxmeyer, the guy who sort of started the use of cord blood stem cells for bone marrow transplantation, did a smart thing when he first banked the first cord blood hematopoietic stem cells. He stored them away in aliquots, and every year after the first cryopreservation, he thawed out a sample and then compared the quality of the thawed sample to the original material. You know what he found? It doesn't change. So 28 years later now, the products that he's thawing today are as good as the products when they were first cryopreserved. So that supports the concept that off-the-shelf inventoriable cells is clearly validated. The other thing is one size fits all. So I mentioned to you before that unique immunologic biorelationship between the mother and the fetus. That means that the placenta and its cells can be delivered to an unrelated, unmatched recipient without the need to do anything. So that's the off-the-shelf, one-size-fits-all concept. And from this approach, we produced a number of products. I'm going to talk briefly about some of them, but the bottom line is we can do everything with cells from the placenta, from treat immunologic disorders, modulate the immune system in autoimmune disease, stimulate repair in degenerative diseases, and specifically attack things like cancer because of those unique immunologic properties. Crucial to all of this, and I'm not going to belabor it because you're going to hear a lot about it, you have to create validated systems that meet what Desiree was alluding to, which is the high standards of the regulatory community. And I'm here to tell you, in our hands, in our laboratories, we produce product that has extremely high purity, extremely high consistency, and meets those high expectations. Now, for all of these things to have a big impact in medicine, you've got to be able to provide access and affordability of products, reliability, and then you have to demonstrate that they work and they work safely. These three pillars of products are met very, very nicely by the platform I've been describing, the placenta, as illustrated here. And right now at Cellularity, which is, I think one of the leading companies in the field, we've got a very, very deep and broad pipeline that is illustrated here that ranges from cancer, hematology and cancer, all the way down ultimately to treating age-related diseases. And I'm not going to bother you with this, but that's about 20 some odd years of work right there. All right, so I want to shift gears a little bit because I said at the beginning that what we originally thought was regenerative medicine is going to be the hallmark and the platform upon which we impact the field of immunotherapy, immuno-oncology, because that right now is getting a lot of airtime. Okay, everywhere you go, you're hearing about patients who are being treated with engineered immune cells that go and hunt down and kill their cancer. That's the CAR T revolution. It's nice to know that that revolution would not have occurred had it not been for all of us working with stem cells. Now, I was talking about the great properties of the placenta. One of them is this immunotolerizing ability that allows the one-size-fits-all. Another interesting property is we recognized several years ago that about 1 in 1,000 pregnant women has some form of cancer during her pregnancy. Okay? I thought that number was surprising to me, but it wasn't that surprising because I've known of situations where mothers, pregnant moms, go to the doctor, find out they've got lymphoma or breast cancer or whatnot, and then the decision is, do I get treated or do I have my baby, right? Well, what's interesting is in the ones who wind up carrying their children, that 1 in 1,000, do you know what the incidence of a mother transmitting her cancer to the fetus is? Doesn't happen. Doesn't happen. It's so rare that the occasional case where someone's trying to say that it occurs winds up being a publishable case report. The bottom line is that we recognize this and decided to chase what might be at work here that's protecting and providing a defense system against the transfer of malignancy. And it turned out that it resided in a natural killer cell. So placental natural killer cells, which can be produced to high quality, off-the-shelf, one-size-fits-all form, meet the anticipated demands of an oncology healthcare system because they can be administered independent as a first-line therapy, but also in combination with other therapies. Natural killer cells are a form of the innate immune system, which is very, very good at specifically hunting down malignant cells as well as virally infected cells and other what I would call serious pathogenic impactors on the developing fetus. So the placental NK cells are there to protect the fetus at all costs. Well, what we simply did was identify the cell and come up with a strategy to productize it and then use it in allogeneic cancers. The first place we decided to take this was in AML, acute myeloid leukemia, a really bad disease if you don't respond to therapy. And what we were trying to do there is boost the response rates in patients who had failed all other approaches. And I don't want to belabor the clinical trial design and all that. Our preclinical data, again, not to belabor it, we obviously saw evidence in the laboratory that our NK cells could work against AML cells. We also found out that part of the way that they do that is by stimulating certain cytokines like interferon gamma, and that they could directly kill AML cells by coming up close and personal, touching those cells and punching holes in them with these molecules called perforins. In our clinical trial, we gave a single infusion of these placental NK cells to patients who had failed pretty much every therapy. These were patients who had a forecasted life expectancy of 30 days. Okay? So this is end-of-the-line kind of stuff. And what we saw in the study was, first and foremost, that these placental NK cells could actually stick around. So a single infusion, these cells actually stayed in these patients. And in about a third of the patients, we actually saw evidence of a clinical benefit. We never expected to see that. This study was designed simply to show tolerability. But in roughly a third of the patients, we got responses or stable disease. And in one case, we were able to clear circulating blast cells from the bloodstream. So the malignant cancer cells were gone. That was a very, very big sort of wake-up call, and it suggested to us that if we treated earlier and treated more often, this would have an impact even in this crappy disease. Well, we took this into the next series of studies to show that not only do the cells stick around, but they remain biologically active, that they actually can mature. And if you look at the lower right-hand corner, we showed that the cells, although they're coming from an unrelated donor, don't do anything to stimulate the patient to respond to them, which means you can give these cells over and over and over again without inducing a rejection of those cells. So in AML, 3 of the 8 patients had evidence of a benefit, and it gave rise to us developing a phase 2 trial design, which is underway right now, where we're actually going to use this product more frequently and early on. And if we can make a similar impact to what we saw right there, this is an approvable pathway for these products. The other place we went to look at was multiple myeloma. As a company that was spun out of Celgene, Celgene is a company that is known for having a huge impact in the treatment of myeloma. We went after patients who were both newly diagnosed and who had relapsed refractory disease. Again, preclinical evidence gave us good support that this made sense. Our study design was that in patients who were heavily pretreated and were failing those treatments, we gave a single infusion of these cells and then followed up. And what I'm here to tell you is that, again, we were very, very compelled by what we saw. 12 out of 15 patients treated had a clinical benefit, either what's called a VGPR, which in myeloma terms means that you're getting a very good response, but you're not clearing the abnormal proteins from the bloodstream, but that's okay. That sometimes takes time. And a large percentage of the patients went into a complete response, meaning they cleared protein and everything. And what was interesting is that in the newly diagnosed population, 9 out of 12 patients being looked at with the most sensitive measure of residual tumor burden had no tumor burden 90 days after treatment. Okay, that's an MRD negative, minimal residual disease negative state. Now that is about 2.5 times what's predicted in that population. So that suggests again that this approach makes a lot of sense. Okay, now I'm going to switch gears and talk about the thing which makes me happy, because as I'm getting older and older and older, I worry about this stuff. Years ago, working with my colleague Craig Venter. Craig Venter, as you know, is the scientist who first sequenced the human genome. He's taught the world a lot about what DNA is. DNA is in essence a programming language. Okay, it's software, and when thinking about it, it dawned on me that if our biological software resides inside the nucleus of a cell, that software has to get read and processed somewhere, and it gets read and processed in the cytoplasm of the cell. That's where the mRNA and the transfer RNA winds up going through the endoplasmic reticulum, and it codes for the production of proteins and so on. But it suggested to me that every stem cell could sort of be thought of as a little computer, okay? With the software in the nucleus, the processor in the cytoplasm, and the surface of the cell membrane acting like a keyboard. And it turns out that this model actually kind of works, because stem cells, when they stick to a surface like a basement membrane, the molecular structure of that surface creates a set of keystrokes on that keyboard that tells the cell where it is. That's why a stem cell from bone marrow or cord blood injected into the intravenous circulation and winds up in the liver doesn't become a bone marrow cell in the liver. It becomes a liver cell. All right? You know, when you walk in the mall and you look at the map, it says you are here, and that's how you find where Nordstrom's is and all that. Okay? The same thing happens with living cells. These cells actually figure out where they are based upon that set of keystrokes. So the reason that was important is that if stem cells are like little computers, and I can figure out a way to capture those computers with their intact, uncorrupted software and store them away, maybe there's other places to apply them. So even though we are living longer and longer and longer than humans have ever lived on this planet, and life expectancy is continuing to move, and it's moving up slowly, the biggest impacts on moving the bar in life expectancy is eradicating causes of premature death. In the early 20th century and late 19th century, it had to do with maintaining higher levels of sanitation and sterility, right? And then antibiotics and other forms of therapy, mostly getting rid of things that killed us by infectious processes. The big impact now on eradicating causes of premature death are early detection of cancer and going after cancer, and early detection of lethal cardiovascular problems, aneurysms, arrhythmias, et cetera, et cetera. But the problem is, as we're pushing the bar up and people are living longer and longer and longer, the price of longevity is that there's an increase in the frequency of chronic illnesses and what I call decline in performance that robs us of the value of those extra years. Things like frailty and neurocognitive decline and so on. Older patients are at higher risk of chronic diseases at alarmingly high rates. And so anything we do to add years to life, I think has to address this. So one of the concepts we had was that maybe there's a relationship that could allow stem cells to find a role in advancing quality and performance as we age. We did a study years ago looking at how placental stem cells could influence an inherited disorder of the skin called epidermolysis bullosa, because we thought that the pro-regenerative Can you guys hear me? Okay. So in that study, let me give you this.
Oh, don't worry about it.
Here you go.
Sorry about that.
No problem.
Still no mic? Still no mic. And now this one's back on. For my next trick.
All right.
Okay, sorry about that.
That's okay. Can you hear me on this one? All right. So in any event, we were looking to see whether we could stimulate repair in this very, very dystrophic skin, and we actually saw that we could. We could actually improve things. But what was unexpected is that the animals who got that improvement also lived longer. So it suggested to us that maybe there's a connection between the stem cells and lifespan. And then while I was running the cell therapy division at Celgene Corporation, because we were doing lots of studies in cancer, and I sat on the executive management committee of the entire corporation, maybe because I was bored or whatnot, I used to look at the data from those cancer studies. And one of the things which I observed, which was very interesting, is that if you look at bone marrow biopsy data from patients in these clinical trials, it struck me that there was a correlation between the number of stem cells in the bone marrow and the age of the patient. Okay? And this graph depicts that this relationship is an exponential decline in the number of stem cells in bone marrow that's age-related. So I said, you know what, I'm going to go and ask a smart guy to help me figure this out if it's true in other systems. So I went to a guy named Evan Snyder out in San Diego. Evan, he was previously at Harvard. And we did a study where we actually looked at the number of stem cells in different organs and tissues as a function of age and showed that this relationship held true, that in fact the number of stem cells declined with age, and that was even associated with a decline in the performance of the organ or the tissue. The obvious thing is, well, if that's the case, maybe I can just add stem cells to an aging system. What would that do? So in the study, in an experimental animal model where we collected stem cells from the placentas at birth and stored them away, and then gave the animals back doses of their stem cells as they aged, we found that the animals who got back their own stem cells lived 40% longer than their litter mates. 40%. Not only did they live longer, but they were bigger, stronger, meaner. We had to separate them because they used to beat up on the little ones. So the truth is that this strongly suggests, and this work's been actually replicated by a number of laboratories, this work strongly suggests that one of the simplest potential ways to augment our health and performance as we age is to simply make sure that the reservoir of stem cells in our body stays in good quality as we age. And there's two things to keep in mind. You have stem cells in pretty much every organ and tissue of your body. The problem is that as you age, you use them up. Okay, you call upon them to renovate and renew your body over time. The other thing is that over time, the existing population of stem cells becomes damaged. Though the DNA in those cells becomes corrupted, it's subjected to viral infections and radiation and chemicals and so on and so forth. So, if you think about it, the physio-anatomic changes that we see as morphing from young to old actually reflects the continual evolution and renovation of those tissues driven by the population of stem cells. When the starting material in those stem cells is of lower quality, the renovation and repair is of lower quality. Anybody ever renovate a house? If you use good new materials, you have a better renovation if you don't. So the preservation, so really one of the things which I'm advocating for now, and as a guy who just got treated less than a week ago, I know it works. I think that for those extra years we add to be meaningful, you have to preserve high-quality human performance. High-performance mobility, high-performance cognitive function, and believe it or not, youthful aesthetics counts too, because it has a big impact on mental status and on the ability to compete in society. So, recharging the regenerative engine seems like one of the easiest ways of going about this. You take stem cells from younger sources, put them into patients who are older, replace their regenerative engine with a younger regenerative engine. I know many of my colleagues here share a lot of these same views. You're going to hear from Professor Hare later on. This represents potentially one of the most valuable ways to deploy cell therapy known in medicine. So, as a company and as a guy who's been leading a group working on turning living cells from the placenta into medicines that can affect a whole range of diseases, we're delivering real paradigm shifts in the way we treat these diseases, and the jury's not out yet on many of the applications, although it's happening literally as we speak. I think in the long run, one of the most exciting opportunities is going to be to use these things to treat age-related degenerative processes and add quality to those added years. Thanks very much.
That was amazing, really. I loved every minute. I'm now a geek for what you do. But I wanted to ask you, you said a couple things that made me wonder about some of the ethics in these things. And one was, I know that a lot of people, including my children, when they have babies, they freeze the cord blood, and they pay quite a bit initially, and then they pay storage forever. But since anybody's can do it when they do the second or third child and pay again for it, is that really necessary, or can one sibling take care of, I mean, how many of them are all going to get childhood cancer, right? So I was just wondering about the fact that so many of us have the impression that we need to preserve each individual's cord blood rather than each one.
That's a great question. So let me try and answer it this way. There is no doubt that there's tremendous effort ongoing to create allogeneic, off-the-shelf products that are available that don't require you to have your own cells banked away to be a beneficiary of those therapies. We're working on it, et cetera. But there is no doubt as well that having your own cells stored at the time of birth that has your specific genetic information in it may afford opportunities to use those cells that off-the-shelf allogeneic might not. That's number one. Number two, the field of cord blood banking, which evolved and started because the hematopoietic stem cells in cord blood was a starting point for bone marrow transplantation, for hematopoietic stem cell transplantation in conjunction with managing cancer and other inherited diseases. And at the time, having your own cells meant a perfect match, which was desirable clinically. But today, what we're recognizing is that in some cases, your own cells might be the best starting material for immunotherapeutic CAR T products and a range of other technologies which will hinge on having kind of your own starting material. But the issue about expense and why bother incurring this expense if there are alternatives, that becomes a personal economic decision, and it's sort of like, you can get around town driving a Subaru. Why do you need that, that Audi Quattro, right? It's sort of like where do you want to invest your money to give yourselves the specific quality of life and lifestyle you want? And the argument that I make to people is this. There's no doubt that there's a cost of banking your child's cells up front. I think the annual storage fees are so inconsequential that few people balk at it, okay? That's my personal belief. And by the way, what's that? Well, but for patients who have clear economic challenges, there are all kinds of benefit programs that are actually providing this sort of free of charge. And that's actually evolving as we speak. And one of the arguments we make is that if you're going to think about the potential impact on overall healthcare costs for that individual for their lifetime, it's a really, really good investment even for government to make at this point, okay? So I think, we should have gotten there by now, but I think we're going to get there in the future. I often tell people, if your baby was born with an extra set of kidneys and an extra set of lungs, would you throw them out at birth? Right? Because again, I actually see these cells as a starting point for making those kind of replacement parts. But the truth of the matter is, cell banking at birth can be offset when the system in place banks some of those cells for the individual and then uses other cells for other people. We're actually pioneering that, okay? It's all about kind of spreading the overall cost of the system over more than one way of paying for it. And believe me, one of the ways to do that is for communities like this to find ways to work together and collaborate so you reach the scale where economics begin to be impacted and you get real economy of scale. So I'm a proponent that every child that's born should have their cells stored away. It's so inexpensive over the long haul. We got to talk to groups like payer organizations and government to say, you know what, if you had this tool available, you may not need to use it in the first 18 years of life, but the probability of needing to use it increases dramatically as they age. And if we can validate this data that says you can add 20 years of high-performance life to an individual, the impact on something like GDP is enormous. I mean, I had an economist do the work here. If, we've moved life expectancy to about 80 years of age right now in the United States. The Social Security system was designed when life expectancy was 65. So the reality is nobody expected you to live long enough to collect anything, right? Today, the healthcare system and the Social Security system is burdened because people are living longer and longer and longer. But if you actually say that retirement, there's no need to retire at 65, and that people can be active contributors to the economic system and to society well into their 80s, that changes the argument as well. But that speaks back to what I was talking about before. In order for that to happen, those extra years have to come with performance, right? So, you know what? Complicated issue. I'm not a politician. We can make some pretty strong political arguments. You just need some time for these arguments to begin to penetrate the forces at control.
I want to say, I mean, this is fantastic. And what I'm getting from the audience is the shifting around a different kind of panel. So what I'm going to do now to continue the strand that is evolving is bring Professor Hare up next to talk about those stem cells that you can get from somewhere else. And then I'm looking at pulling together We have the California Cryobank here with Jamie and Kate Brown to have a combination panel with all of you here so that we can open that up for your questions and start to look at collaboratively what we can do to move this along and make this kind of thing more available in lives. So thank you as usual. Fantastic, Bob. All right.